Thank you for joining me today. I'm Dr. Gangat, and today I will be discussing the mechanism of action for DISC-0974. DISC Medicine strives to develop hematology-focused treatments that will have a meaningful impact on patients' lives. DISC-0974 is an investigational monoclonal antibody against hemojuvelin in development for the treatment of anemia of inflammation. Clinical trials are ongoing in myelofibrosis, chronic kidney disease, and myelodysplastic syndrome. DISC-0974 is an investigational medicine and is not approved by any regulatory authority for any use. The safety and efficacy of DISC-0974 has not been established. Iron is essential for normal red blood cell production and oxygen transport. Disruptions in iron metabolism and utilization can therefore lead to decreased hemoglobin levels and anemia. Iron concentrations in plasma are maintained through a complex feedback loop and controlled by the interaction of the peptide hormone hepcidin with its receptor and cellular exporter ferroportin. Ferroportin is found in iron-storing and iron-transporting tissues and is needed to deliver absorbed dietary iron to the circulation from sources such as duodenal enterocytes, macrophages in the liver and spleen, and iron-storing hepatocytes. Iron is transferred from the duodenum into the blood, then transported by transferrin to cells or bone marrow. Hepcidin regulates systemic iron levels by binding to ferroportin, causing cellular ferroportin internalization and degradation. In the liver, hepcidin regulates mobilization of iron from hepatocytes and iron recycling of red blood cells in macrophages. Anemia of inflammation develops due to a dysfunctional increase in hepcidin caused by chronic systemic inflammation or inflammatory stimuli. Increased hepcidin contributes to anemia primarily by reducing the intestinal absorption of dietary iron, preventing the release of recycled iron from macrophages into plasma, and reducing the availability of iron stored within cells of the liver. Control of hepcidin activity may have therapeutic benefit in the treatment of iron-related diseases, including anemia of inflammation. Hepcidin is regulated by the interaction of several signaling factors. Hemojuvelin or HJV is a regulator of hepcidin production and functions as a co-receptor to the BMP signaling pathway. HJV complexes with BMPs, resulting in the translocation of SMAD proteins and the stimulation of hepcidin production. High levels of HJV increase BMP signaling, causing hepcidin overexpression and low levels of iron in blood plasma. Conversely, when HJV activity is inhibited, BMP signaling is reduced. DISC-0974 is a pathway-specific anti-hemojuvelin antibody designed to reduce hepcidin levels by binding to HJV. Reduction in hepcidin concentrations result in increased iron availability and release of stored iron and may increase hemoglobin. In non-human studies, inhibition of HJV activity following inflammation prevented hepcidin increases and the decreases in serum iron associated with anemia, demonstrating the potential for HJV as a target in treating anemia of inflammation. Thank you for your time and interest in this presentation of DISC-0974.